Well, hello, welcome to the Broad Institute, and thank you for joining us for our brand new public lecture series called Science for All Seasons. Probably many of you have been to visit the Broad before for our public lectures in the summer, and at the last lecture of the summer I asked the audience whether they wanted us to extend these lectures into the academic year, and there was this, every hand went up, and so we're going to try it. You're our experimental audience. We're trying different topics. We're trying different lengths of time. And the only way that we can make this series what you need it, what you want it to be, what you most want it to be, is by telling us. So I hope you'll give me feedback, catch me at the reception afterwards, and let me know what you'd like to see and what days work best for you or whatever. So as you know, for all our lectures, as with any of our public lectures, Science for All Seasons is our chance to brag about the research that happens at the Broad and enable you to explore these hot topics in science that are happening here. Like Midsummer Night's Science, it's free, it's open to the general public, and that includes students for all ages, so welcome, the crowd that just arrived, our neighbors, our colleagues in Kendall Square, anyone with an interest or curiosity about science. I hope you find tonight interesting. I hope you come back. I'm going to tell you about two other events, one here and one at our partner at the Koch Institute coming up over the next month. Our next lecture will be November 7th. It will be Romnick Xavier, and he'll be talking about guts and bugs in health and disease. And across the street at the Koch Institute on October 29th, there'll be a panel discussion called Women on the Front Lines in the Fight Against Cancer. So if you're interested in going to that, just go onto the Koch Institute website. Their lecture series is called With Insight, and you're welcome to join them there. Also, as many of you know, for those of you who can't join us in person, we record our public lectures and make them available as soon as possible after the event on our website and on YouTube. And so if you're interested in knowing about the science that goes on around here, you can subscribe to that YouTube channel and see any of the lectures we've already given. If you can't make a day, you can watch them there or share them with your friends. So we're really excited to be starting this series in the academic year, and we're really excited to have Elinor Karlsson being our kickoff speaker for this event. Elinor is a postdoctoral scientist at the Broad Institute, and that makes you think she's only been at the Broad for a little bit of time, but that's actually not true. She's one of those people who first started working at the Broad before it was a Broad. She started working with Broad Institute scientists in 2002, before she started her graduate research. She's done most, if not all of her research here at the Broad, even though she was a student at BU, and she's now a postdoc in Pardis Sabeti's lab, and so is officially affiliated with Harvard University, although I see her here all the time. She's really interested in looking at evolutionary history, and she combines tests for natural selection with genome-wide association studies to identify genes, pathways, and functional variants underlying polygenic disease. She's currently using those approaches to find the underlying genetic causes of obsessive-compulsive disorder, using dogs as a model organism, and also cholera resistance in humans, and she's going to talk about her OCD work today. So please join me in welcoming Elinor Karlsson.
Thank you very much for the introduction. Thank you to all of you for coming. I'm really excited to have the opportunity to talk here. My title says I'm going to talk about OCD, which I will, but I've got a bit of background first. It's kind of a wider scope talk. But first, I just want to thank our research collaborators on this project. All of our work is done using blood samples from pet dogs, so we're very reliant on dogs and their owners contributing to our research. And I'm going to go back to the dawn of the genomics era, which was probably about 2000. Well, to be fair, it was probably more like 2000, but it was a long time ago in genomic time. And we decided to sequence the genome of the dog. And so just a reminder for those of you who don't know, the genome is basically, all it is what we call the DNA that you've inherited from your parents. So you get one copy of the genome from your father, one copy from your mother, and then you carry a copy of your genome in every single cell nearly in your body, which is why we take a blood sample and we can get your DNA out of your blood. And so what we did was we sequenced the genome. So we did the DNA that was in the cells of a dog named Tasha, who was a female boxer. Her genome was about 2.4 billion bases long. So those are the letters of DNA, A, C, G, and T. This was pretty much comparable to humans, which is 2.8, and mouse, which is 2.6. And we used Tasha as a starting point and then looked at a bunch of dogs from different breeds and started to look into the history of dogs and found out that was actually kind of cool, is that dogs were actually domesticated from wolves probably somewhere between 10 and 30,000 years ago. We don't know why, whether they decided to start eating our garbage, whether we decided to start adopting wolf pups because they were useful for guarding, but that was about when it happened. And then much more recently, for the most part within the last 200 years, modern dog breeds were created. And so these are all the various, pugs and chihuahuas and Great Danes and everything that you see running around today. And if you actually look at how different a pug and a chihuahua are, and in this case a Great Dane and a chihuahua, they're about 99.9% similar, which is very similar to humans, one change every 1,000 bases. The big difference here is that within the Great Danes, all the dogs are very related to each other, so they're essentially all cousins of one another within a dog breed. And if you actually look at what's going on in the genome, I'm just going to make this point because this comes back several other times in this talk. So the dog genome is 2.4 billion letters long. We usually think about genes when we think about the genome, but in fact, the genes only make up 1.5% of those 2.4 billion bases. Those are what we call the coding parts of the genes. These are the ones that you might remember from school as the parts that get made into proteins. But there's a lot of other stuff going on in the genome. So you've got your gene, and the gene actually has these parts that don't go in the protein called introns. You've got regulators and insulators and enhancers and things that basically control when the gene gets turned on and off and where it gets turned on and off. And we basically need to find out where else in the genome is important outside of the genes. And one way that we do this here at the Broad is we actually look for what we call conserved DNA. And so the theory here is 2.4 billion letters long. Every time you pass your DNA on to your children, random changes happen in it. And over time, random changes will accumulate in the population everywhere in the genome. It's copying itself. It's got 2.4 billion letters to go through. It makes a few mistakes. However, if that piece of DNA is important, if it's doing something critical to the function of the organism, it can't be changed. And when changes happen in that piece of the DNA, the animal will die, and so it won't get passed on. And so we actually can find the important pieces of the genome by looking in dogs and humans and mice and bats and elephants and rats and saying, if there's a piece of the genome that's almost unchanged between all of these species, it's probably doing something important. And so that's what we call the conserved DNA. And so we looked at that and we said, oh, great. 5.5% of the genome is either coding or conserved, which is important as far as disease goes because most of the disease-causing changes or variants in the genome do not actually happen in genes. They're what we call non-coding in the regulatory region. But it also brings up the point that I'm going to have a job for a long time because we still don't know what most of the genome's doing. So this is the start of the whole story, and we decided to go, can we find genes in the dog genome? And so what are we doing here? Basically, this was the same thing we do in humans when you talk about human disease studies. And what we're doing is we're taking a bunch of healthy dogs that we call controls. We take a bunch of dogs that have some kind of disease or trait, in this case, white coat color, and we say these are our cases, and we look at their genome and we compare their genomes and we say, where are they different? And we do this with something called a SNP chip. So a SNP is a single nucleotide polymorphism, which is a fancy way of saying a one base change in the genome. And so this is a SNP, so you can see this dog's got two A's, one A from each parent here, and this dog's got two C's, so there's a difference at these positions. And rather than looking at all 2.4 billion letters of the genome, we found a bunch of positions that we knew varied between dogs, and we just focus on those. And so this is about one SNP every 10,000 to 10,000 bases when we use a SNP chip. The one we use for dogs right now, our array that we use to test SNPs, is what we call low density, so it's probably more like one every 10,000 bases. And so when we talk about our working with purebred dogs, that's kind of a key point right now, is that the reason why we work with purebred dogs is that the array that we're working with works best in purebred dogs. But now, just in the past couple of years, it's gotten much, much easier to do what we call sequencing. And what sequencing is you're just looking at every single letter in the genome. And the nice thing about this is you can find positions that have changed that you didn't know to look for. Problem with it is that up until now, and still going on, it's still a lot more expensive than doing a SNP chip, but this probably isn't going to be true for very long. And so we got our SNP chip and we got our dogs, and we're going to look at where the disease variants are. And so these are all the SNPs, and all we're looking for, it's actually a very simple test. We look at a whole bunch of solid dogs, a whole bunch of white dogs, and we look for the positions that very strongly correlate with the trait that we're interested in. We say, this is probably sitting near our disease variant or our trait variant. And then we go and sequence this piece of the genome and look for the changes that might be causing our trait. Because with the sequencing, we can look at all of the bases. And so basically, when we're looking for disease-causing mutation, we first start by using a SNP chip to look at the whole genome. This finds kind of the neighborhood that the mutation is in. So it's about 1 million bases usually we can get down to in dogs. We then sequence that region to find the actual candidate mutations, the things we think might be causing our trait. So we usually get 10 to 100 out of that. And then we have to go to the people in the lab because we have to figure out which of those 10 to 100 is actually causing the trait, and that's very hard. And so this is kind of where things tend to get slow these days. But we can get pretty close, even with just the SNP chip and the sequencing. And so we've started this a few years ago in dogs. And so I'm just going to quickly give you a couple of our early disease mapping stories that I think are quite a lot of fun. So we've got, let's see. So the first one is what I would call easy-ish. It did not seem easy at the time, but in retrospect, it seems like it was easy. And so this is the hairless trait in the Chinese crested breeds. And so if they get just 2 normal versions of the hair gene, they're what they call coded, so they look like this. If they get 1 bad version or 1 non-functional version of the hairy gene, they look like this. They have no hair. They have very little hair. They have really bad teeth. If they get 2 copies of the gene, of the hairless gene, 1 from their mother, 1 from their father, you'd never see the pups because they die in utero. They're not born. It's a lethal mutation. And so we took this trait and we mapped it across the genome. And so all you're looking at here, so this is the whole genome. Humans have 23 chromosomes, dogs have 39. So each of these is a chromosome. Each of these little dots is a SNP. And all this is showing is for that SNP, how different are the hairy dogs from the hairless dogs. And so the higher you are on this axis, the more different the 2 groups are. And you can see quite obviously, we have a really nice peak there. We looked under that peak. It was over a gene called FOXI3. Nobody knew a whole lot about that gene, but we went ahead and sequenced it to see if there's anything going on in there. And what we found was that sure enough, this is what our hairy dogs looked like. 1 copy of the DNA from their mother, 1 from their father. This is the hairless dog, and basically they took a piece of their DNA and it was duplicated, so it's 7 bases. And the effect of that 7 bases is that your protein gets totally messed up, because for those of you that kind of remember the whole 3-base codon thing, your codon chain is all shifted off by 1, the protein truncates earlier, and basically it just can't do what it's supposed to do. And they actually went, okay, we don't know a whole lot about this gene, and they looked at mouse embryos and found out that it is indeed expressed in the hair follicles. You can see here these little black dots are where that FOXI3 protein is getting made, and these are the kind of beginnings of whiskers in a mouse embryo. And so we thought, that's a pretty cool story. And then we went and looked into 2 other hairless breeds, the Peruvian hairless and the Mexican hairless. And we found out that not only do these breeds have exactly the same hairless trait, they actually have exactly the same mutation in their DNA. And if you think about it, 2.4 billion letters of DNA, what are the chances that you're going to copy exactly those 7 letters over again? You might come up with another way of breaking the gene, but you're not going to find that exact mutation unless it actually only ever happened once. And all of these breeds have inherited exactly the same hairless mutation, which actually tells you how old it is, because you got a Chinese crested, which, Asia, and Peruvian hairless and Mexican hairless, New World, Americas. So this is a mutation that basically dates way back in time. And so that was kind of a cool story. So that was great. As I said, we actually hit that 1.5% of the genome that makes coding, codes proteins, and it was fairly straightforward. But then we moved on to something a bit harder, which was coat color in the boxers. And so if you get 2 solid genes, 2 copies of the solid version of the coat color gene, as we call it, you're solid. You don't have hardly any white on you. If you get 2 copies of the white version of that gene, you get to be totally white nearly. Sometimes you have patches on your head. If you get 1 copy of each, you are flashing, which means you're half brown and half white. Very conveniently, this happened to be what the dog that we sequenced looked like. So we knew that in our genome data, that for the whole genome sequencing that we did in 2005, we had to have that dog had 1 copy of the white mutation and one copy of the white version of the gene and 1 copy of the solid version of the gene, which meant that we kind of had sequence data already, which when we were doing this project a couple of years ago was a big deal because sequencing was hard. Sequencing has gotten much faster and much cheaper recently. And the other interesting thing about this trait is that out of the white boxers, about 2% of them are totally deaf. And so there's some connection between your skin color and deafness. So once again, we mapped it with a SNP chip. We got a really strong peak. We looked at that peak. It was over the gene MITF. We sequenced MITF. We looked in the coding regions to make the proteins, and we found no changes at all. So that was great. we didn't know quite what to do then, because once you move out, you find a mutation in the piece that makes the protein, it's pretty easy to guess that it's going to break the protein and do something. But here, we've got a whole lot of other stuff. But we thought, OK, we've got another breed here that actually has the same trait, the bull terriers. They also come in white. And we looked in their sequence along this region as well, and we found out it's actually not 1 million bases. There's only 100,000 bases where these two breeds are exactly the same. The white dogs in these two breeds are exactly the same. 100,000 bases sounds like a lot, but when you're starting from 2.4 billion, getting down to 100,000 is actually pretty cool. So we looked at this and we went, all right, cool part of the gene. So it turns out this gene actually comes in at least 2 different forms. So the bars here that I'm showing are the parts that get made into a protein. So those are the coding parts. You can start making your protein at A, which means you have all of these bars going into your protein. Or you can start making your protein at M, which means you start it right here and you only have these pieces. Now, it turns out that if you start making your protein here at M, you actually make a protein that's absolutely critical for pigment cell migration. So it turns out that when you're during development, your pigment cells, they actually originate on your neural crest, so along your spinal column very early in development, and they originate there and then they migrate. So they basically spread around your body and eventually meet up at your stomach, and then you're got pigmentation all over you. Well, as this flashing box here is quite clearly showing us, in the dogs that have 1 copy of the white mutation and 1 copy of the solid version and the white version, they have basically impaired migration. And so their pigment cells are migrating, but they're only getting halfway around. And so it does indeed look like we have some kind of mutation that's affecting pigment cell migration. And so that's great. We sequenced the region. It turns out that in that 100,000 bases, we had 124 candidate variants, so differences between the white dogs and the solid dogs. 48 of those we never saw in breeds that never came in white, so that's all of these other breeds. And then out of those 48, 2 of them occurred in parts of the genome that were really highly conserved, so these parts that never changed between species. And so these were our top 2 candidates. It's what we call a sign insertion, which is a 200 base piece of DNA that got inserted right here. So the white boxers have a piece of DNA, 200 base piece of DNA put right here that the solids don't. And then they have a slightly longer version of the DNA here compared to the solid dogs. And so here's the M start site. All of these boxes here are places in the genome where other proteins come in and bind it to tell you whether to turn that gene on or off. Probably what's happening is in these white boxers and bull terriers, that gene's not getting turned on properly, so the pigment cells are not migrating properly. And it turns out that in order to be able to hear, you have to have pigment cells in your ears. And so in 2% of the white boxers, their pigment cells don't even get as far as their ears, and that's why they're deaf. But the other really cool thing this brings up is why dogs are a great model for human disease, is because you could go and break this protein, MITF it's called, in mice, try to figure out what it does. And what happens is you get white mice, but they're also completely deaf, they're also completely blind, their eyes don't develop properly, and they're also dead. So very bad protein to go and mutate. And you may not actually learn a whole lot about how that protein functions based on the fact that you've got this very extreme version of the trait. But in dogs, we have a naturally occurring mutation. People would not have wanted dogs around that were white, deaf, blind, and dead, so they wouldn't have kept that mutation. And so instead, we have dogs that are white, but rarely deaf. They have normal eyes, and they're not dead. So this is actually really cool, because rather than just finding the coding mutations in that 1.5% of the genome, using dogs to map traits, we can actually find regulatory mutations, which are probably much more representative of the kind of mutations we'd expect to see in humans. And so this was great. These are both single gene traits. 1 gene, you change it, you get the trait. We did this with a bunch of other traits as well. There's an ALS version. There's an ALS that shows up in dogs. There's a ridgeback, the neural tube defect, a bunch of different things. We're like, great. We can map single gene Mendelian traits. But that was kind of just the beginning of it. I was totally looking at the screen for my time, and now it's gone dead. But just going to check my phone to make sure I'm not off by too much. All right, so single gene Mendelian traits, but brains are really complicated and they're inherited. So they are genetic, but there's many, many genes involved in the way your brain looks. And so can we actually use dogs to help us figure out the function of the human brain? So we actually address this in 2 ways. In the OCD work that I've been doing, we've been working within breeds, so that's kind of post-breed creation. But we also have this really cool thing in dogs, which is dog domestication. And clearly, dogs don't behave like wolves. And so when they domesticated them, they changed their behavior, they changed their appearance, and there's probably other things that they changed as well. And what we can do, and this is actually a project done with our group in Sweden a couple of years, that actually just came out earlier this year. And what you can do is sequence wolves, so get the sequence of all the DNA in a bunch of wolves, so 12 in this case, and then get a sequence of all of the DNA in a bunch of dogs. And look for places in the genome where the dogs are both different from the wolves, because it's a domestication locus. The wolves won't look the same as the dogs, but all of the dogs look the same, because all of the dogs are domesticated. And so this is exactly what we did. This very complicated looking plot is just two different ways of measuring it. This measurement is the dog-wolf difference, so the higher you are here, the more different the dogs and the wolves are. And this piece down here is the dog similarity, so the further down it goes, the more similar all of the dogs are. The first thing you can see from this, as I said, is that things get way more complicated than with those single gene traits. It gets much noisier and harder to pick out what's actually going on. But what we saw is that if you actually look in the top peaks, 19 of those peaks are containing brain genes, which is actually very interesting. We know their behavior is different. And if you actually look at the brain genes, they're involved in learning and memory and things like that. And so we actually suspect that this probably has something to do with the reduced aggression and altered social cognition in the dogs. So you probably would not want a wolf that close to your baby. But the other thing we found, which I think is really cool, because it kind of illustrates with this whole looking at the whole genome approach, you can find things you didn't actually ever think you were going to be looking for. And so we also looked in those peaks and found out that a lot of them actually contain genes related to digestion. And when you think about it, this makes sense, because wolves tend to be carnivores. They eat a lot of meat. They're hunting. The big thing that dogs would have had to change in order to live with humans would have been their diet. All of a sudden, they're eating what we eat. They needed to become omnivores. And so you basically have massively upped the ability of these dogs to digest starch. And it's actually a super cool story, because if you, out of those 10 genes, there's 3 of them, these 3 here that I've shown in red. So here's your starch. One of these genes, AME2B, actually makes an amylase, which changes starch to maltose. Dogs, compared to wolves, have 3 to 16 extra copies of this gene. So it's just been copied over and over and over again to improve the ability to digest starch. Then you have to change your maltose into glucose. Once again, we see a gene here, mGAM, which is 12 times upregulated in the dogs compared to the wolves. And then finally, you have to get your glucose into your cells. And we find that there's a protein that's involved with actually transporting the glucose, where the protein structure has been changed in dogs. And so we weren't looking for digestion genes, but we totally found them, and it actually fits perfectly with what you'd expect to see. The Broad Communications people came up with this picture. I think it's super cool. So you've got human agriculture tying into dog domestication. Dog domestication, we then start selecting for behaviors like hunting behavior and herding behavior, but then we also started creating breeds, and a lot of the breeds actually have trouble with problems with neurological disease, and all of these things are tying together. And so another way to think about this is we have these dog breeds, and lots of them were selected very strongly for particular behaviors. Your golden retrievers retrieve, your border collies and your sheepdogs herd. With a lot of dogs like the Afghan hounds that were bred for speed and liking to run. But at the same time, when they create these breeds, the way you create a dog breed is you take your dogs and you basically select out a small number of dogs and you make those into your breed. And as I said, they're all very related to one another. And if you happen to select a gene that causes or contributes to a risk of having a psychiatric disease, then a lot of the dogs in that breed are going to have that psychiatric disease. And so that bottleneck, as we call it, where you go from a big population down to a little population, has a side effect of increasing rates of disease quite often. And so you'll see that particular neurological diseases are actually enriched in particular breeds. And so you'll see noise phobias, anxieties, there's breeds that get epilepsy, dominance, aggression problems. And a lot of these diseases in dogs actually have a human correlate. So the canine compulsive disorders, they call it like human OCD. You've got things that are like Alzheimer's disease as they get old, impulse control disorders, panic disorders, separation anxieties, and all of these have great models in humans. And OCD in particular is one of the best models of a human psychiatric disease in dogs. And so OCD is characterized by a normal behavior that's done way too much. In humans, that might be hand washing. In dogs, it might be tail chasing. That behavior is distressing, time-consuming, and impairing. You're doing it so much that it impairs your ability to live a normal life. In humans, they're often completely aware that what they're doing is irrational, but they can't stop themselves. And the onset is often very early. So in humans, the most severe forms of OCD often have onset during adolescence, and similarly in dogs, you're seeing it usually somewhere between 5 months and a year. And we actually treat the dogs and the humans with exactly the same drugs, often these what we call selective serotonin reuptake inhibitors, or SSRIs. And in both species, they don't work that well. Maybe 50% of patients even get a response. And so this is a disease for which we desperately need improved therapies. And in both species, it's a very strongly inherited disease. So in humans, it's estimated to be at least somewhere between 45 and 65% genetic. And in dogs, it's hard to actually quantify, but we know that particular breeds are much more likely to get OCD, which is an indication that it's genetic and not environmental. But there are differences. As I said, in humans, it's 1% to 3% of people. It's a very genetically heterogeneous disease, so a lot of different genes are contributing, which makes it hard to find any particular one. But in dogs, we've got it very common in some breeds. Humans, it's characterized by both compulsions and obsessions, so worrying about whether your house is locked or whether something bad's going to happen. In dogs, it's characterized by compulsions. To be fair, we don't know whether they're obsessing. They don't talk. They could very well be obsessing. And of course, the behavior differs. So in humans, hand washing, checking the locks, hoarding. And in dogs, it's tail chasing, sucking, and fly snapping. And so this is a video taken by the person that actually worked with us on this project, Alice Munfinelli, showing a bull terrier spinning. And so they'll do this for hours and hours on end, just chase their tails. And then similarly, the trait that we actually worked with in our genetic study was in the Doberman pinchers, who do this thing that they call flank sucking. And so they'll basically just turn around and gnaw on their bodies for ages. And so what we did is we took 92 Doberman pinchers that had this trait, and we took 67 Doberman pinchers that didn't have it. As you can see, it's quite early onset. They have two different forms. Blanket sucking is considered slightly less severe, and flank sucking is considered a little bit more severe. This is clearly flank sucking. But the blanket sucking dogs often will end up consuming blankets, ending up in the hospital, having to have surgery. It's a pretty awful disease. And so we did our same thing. We mapped it with a SNP chip, but as you can see, as I was mentioning with the domestication, it's a much noisier signal than what we saw with the single gene traits. But lucky for us, this is a particularly successful project. We had one peak that really stood out. And under that peak was a gene called CDH2. And the other really cool, interesting pattern we saw was that if at that top SNP, so the one that's right at the top of this peak right up here, if you had a T at this top SNP rather than a C at the top SNP, it meant you had a higher risk of disease. And so what this means is, so the unaffected dogs rarely had two copies of the T, so getting one from each parent. They occasionally had a CT, but mostly they were CC. They had two Cs, one from each parent. But blanket sucking dogs, the less severe form, were more likely to be TT. Flank sucking were even more likely, and then the multiple behaviors, ones that were doing both blanket sucking and flank sucking, had the highest rate of the TT and the lowest rate of being CC. Very few of them had two normal copies of whatever was in that region. And so that was actually exactly the pattern we would expect for what we call a risk locus, or a place that's correlating with behavior with the trait. And so CDH2, so we were very excited when we found this, because it's always much easier when your gene is exactly the kind of gene you would expect to find. CDH2 is a neural cadherin. It's found in something called the glutamatergic synapses, which was immediately kind of interesting to us, because if you remember, we treat OCD right now with selective serotonin reuptake inhibitors, and they don't work that well. And the fact that we have something acting in the glutamatergic synapses, so things that respond to glutamate, might suggest that we're actually targeting the wrong neurotransmitter there. It's involved in synaptogenesis, making new synapses, and also synaptic plasticity, which is basically a fancy way of saying learning and memory, making new synapses in order to store new information. And what it basically does, here's the one side of the synapse, here's the other side of the synapse. This is synapse, this is the cadherin right in between in red. And basically it sits there, when it binds calcium, it gets rigid, and that rigidity kind of sticks around for a while, and is a way that connections in the brain kind of get stabilized while they get kind of set in, it's not a permanent connection, but it's kind of like scaffolding while you build up that connection and make it stronger. But we thought this is great, but yes, this is noisy. What about all of those smaller peaks? We don't know exactly what's in those peaks yet and why they're coming up kind of strong. But we do know that our top locus is only explaining 5 to 10% of the risk. That's this red part here. And the other ones that we found in the genome, the top ones, seem to be getting us up to about 50% of the risk. So that's pretty good. Compared to humans, it's way higher than we'd ever see with a human trait. But we still have an awful lot of of stuff that's unexplained. And this could actually be either risk variants that aren't tagged by the SNP chip, because I said it's quite sparse. We may need sequence data to find them. But it also could be non-genetic factors, environmental factors influencing the Doberman's risk of having the trait. But we do see patterns. We do see that in those other peaks that we find, we see more glutamatergic synapse genes, and we also see more genes involved in learning and memory. And so we're actually very excited to start figuring out more about what's in those peaks. But of course, the other thing that comes in here that we were kind of asking about was that, this is great. We found the things within the Dobermans that are different between the Dobermans that have OCD and the Dobermans that don't. But there's also a chance that because of some kind of selection for behavior, all of the Dobermans are at an increased risk of getting OCD. Maybe they were selected for some kind of, I don't know, chasing or something in the past where they had to worry a whole lot about, chasing something down. And that inclination to be very concerned about something is part of what contributes to the risk of getting OCD. The problem here is that because it was selected in the past, because all of the Dobermans were kind of chosen because they had that trait, we don't have any controls. We don't have any dogs in the breed that don't have that trait. And so we can't compare cases to controls and find differences. Instead, what we did is we looked for places in the genome where all of the Dobermans looked the same, so parts of the genome that seem to characterize Dobermans, and they were also, for the most part, different from other dog breeds. And as I said, this is also something we're still working and digging into, but we found some really interesting genes that we haven't totally proven yet, but we're very excited about them because we found both genes that regulate CDH2, so decide whether it gets turned on or off, and we also found genes that are basically proteins that anchor CDH2 to the membranes at either end, so basically help control how stable that connection is. And so what we're doing right now, as I said, we want to look into these in more detail, so we're going to be sequencing both the genes that were associated, so the ones that were different between the cases and the controls, and we're also going to sequence the genes that were in these selected regions and look in them and see whether there's any changes in them that seem to be connected to OCD. And so the way we're doing this is rather than just looking in the Dobermans, we're going to go to other breeds that have OCD as well, because you might think that if a mutation causes OCD or is partly responsible for causing OCD, then you would expect to see it in some of the other breeds that have OCD as well. And so we've done that now. We've sequenced all of the top regions. Problem with the genome, it's really big. So we've actually got 30,000 variants that we're looking through and trying to figure out what they're doing. So I'm not going to go into all 30,000 in any kind of detail here, but I will say, coming back to that whole conserved thing I came up in the beginning, we see one very interesting pattern, which is that if you look at all of our variants and see where they sit in the genome, if you look at, so basically the red here are variants or genetic changes or mutations that we see only in dogs that have OCD. And in blue are the variants that we see only in dogs that don't have OCD. So you would expect that the mutations that are causing OCD are going to be in the red box. And if you look at how many variants are found that are only in the OCD dogs or only in the healthy dogs across the whole genome, it's basically the same. But if you look in the places in the genome that are coding and conserved, so this is the 5% of the genome that we think is actually doing something really important, we actually start to see a difference. And we see the same thing if we exclude the genes and just look at the conserved regions. And so this suggests to us that we are finding some interesting variants here that we need to start figuring out what's going on. And so why are we doing all this in dog? We still have a lot of questions. We don't have a lot of answers. We're still trying to figure out exactly what's going on outside of CDH2. But this also comes back to the question of what's going on in humans. Is dog adding something to this whole puzzle? And so this is our genome-wide association study that I just described to you, the one that found CDH2 in dogs. We had 90 Doberman cases, 67 controls. In that study, we had about 50,000 SNPs. We had one significant peak, a bunch of interesting things, and under that significant peak, we had one gene, CDH2, which is now a very strong candidate for being involved with OCD. Just a couple months ago, they published the first genome-wide association study in human patients. They had 1,465 cases. They had about 6,000 controls. They tested nearly 500,000 SNPs across the genome, and they didn't find anything. And this is why dog breeds are super cool. Just want to put that out there. But there is something interesting there. So even though none of them was significant, and so when we say significant, we mean that we basically have this threshold in science saying that everything that we call real has to have less than a 5% chance of being wrong. So we have to be 95% sure that we're right. And so this is basically, they don't have anything exceeding that 95% threshold, but they have things that are getting close to it. And one of their top genes is a gene called DL-GAP1. If you actually look at this, so this is back to our synapse again, your pre-synapse and your post-synapse. This is your cadherin that we found in dogs. And this is the one, CDH2. We know that it's turned on in glutamatergic synapses. And we also find that this gene DL-GAP2 is also involved in glutamate signaling, and it's basically a protein. It's also called SAPAP in mouse, so some of my figures say that, but you can read that as DL-GAP. I don't know why they have different names. But it's involved in anchoring those cadherins to the membranes. And they actually found a bunch of other glutamate signaling genes as well. And in fact, another gene, DLGAP3, this is what happens if you break that gene in a mouse. They basically get something that looks an awful lot like OCD. So these mice groom so much that they end up getting sores on their faces. It's really quite sad for them. And so this was published in 2007, and what they saw is not only did these mice have OCD, they also had an anxiety disorder, even though they were just as active as a normal mouse. So the mouse without DLGAP3 is in black, and this is the normal one in white. So they're just as active, but they don't like to go into the light version of the light chamber of the, they got a dark chamber and a light chamber, and they also don't like to move between them. And so they tend to spend most of their time in the dark. And if you treat them with selective serotonin reuptake inhibitors, very much like the humans, especially for the grooming, the OCD-like behavior, they get better. They go from the white bar as far as how much they're grooming down to the black one, but they're still much worse than a normal mouse. However, if you take, and this is work you can only do in lab mice clearly, but you can actually take DLGAP3 as a gene and in an adult mouse put it back into the brain. And they tried putting it in a bunch of different parts of the brain and it didn't really help. But if you put DLGAP3, put a functioning version of it back into the striatum, the mouse stops grooming and their sores heal up. And so you can actually fix their brain, basically. And so that was key in telling you that, so far we've got glutamatergic synapses and glutamate signaling being really important, and it being really important in the striatum. And it basically ties into what they call, let's see if I can get this right, cortico-striatal-thalomo-cortical circuit, or the CSTC circuit. It's a very fancy way of saying is it's this signaling pathway in the brain. You take information in the cortex, it signals the striatum, which is what's responsible for telling you what kind of behavior you're going to do, and then the striatum tells the thalamus about what it's done, that feeds the information back to the cortex again. And very recently, just a couple months ago, there was some very cool work published where they have a new way of using light to turn on brains, and they basically turn on genes in the brain of the adult mouse. And so it's kind of like putting the DLGAP3 back in, but much more targeted. And so what they actually found with this circuit is that if they turn on those glutamatergic neurons in the cortex, so these are the neurons that are signaling from the cortex to the striatum, if they turn those up, they get mice that groom, and they actually groom for up to 2 weeks. And so it's actually a slow onset, but they actually get an OCD behavior. Similarly, if they then so this cortex is signaling the striatum along these glutamatergic neurons. If they inhibit the response that the striatum's giving to those signals, they actually get these mice that have better control of conditioned responses. And so in this case, it's a drop of mouse, they trained the mice, that a drop of water would fall and then they'd have to clean themselves. And they paired it with a tone, and then they stopped bothering with the drop of water. And most mice would learn, the tone doesn't mean anything anymore. But the mice that had OCD would just keep on grooming themselves. They just couldn't stop. They'd been taught to do it and they couldn't stop. But if they actually inhibited the response along the circuit, you managed to control that response. And so the mice are actually telling us that what we've been learning from the dogs and the humans fits perfectly with what we see in the mouse brain. And so it looks like this synapse, this circuit, the glutamatergic synapses in this circuit are a common mechanism in dogs, humans, and mouse OCD. And so now we have this idea that we're going to design, so basically we can use these species together, all the advantages, dogs as a natural model for humans, humans being the ones we're actually worried about mostly, although actually I'm quite worried about the dogs as well, and the mice, which are a great laboratory model. We can do things with them that we can't do in the other species to actually figure out exactly what the mechanism is that's underlying OCD. And the idea here is that if we could actually target exactly the mechanism in the brain that's causing the disease, you could develop therapies that both were much better at treating the disease, but also didn't have the side effects of current psychiatric drugs. And they can help us understand OCD. They can help us understand. So it turns out autism, a couple of the genes that have been linked in humans are also cadherins, so CDH9 and 10. And these two diseases are often linked in humans, comorbid anxiety disorders, and possibly also schizophrenia. It seems really different, but there's a paper out about schizophrenia that said neuronal calcium signaling is really important. Cadherins are activated by binding calcium, so there could be a link there. And so finally, I just want to end by thanking, this is work done by a huge group of researchers, and I can't thank all of them individually, but I just want to point out, Kerstin Lindblad-Toh leads the group here at the Broad. Hyunji and Ruchi are two postdocs that did a lot of the work and analysis on this project, as well as Guoping Feng over at the McGovern. And Alice Moon-Finelli, who's actually here tonight, is the one that did the phenotyping of the Dobermans, which was, I just want to point out, kind of the beginning of all this work. It's defining the cases and the controls that was the start of that. And everybody always asks me whether I have dogs. I don't have dogs. But I do have two really cute cats, so I put them on there. And then finally, I just figured I'd put this out there. If you have a purebred dog and you'd like to help us, we're always looking for blood samples, and we have a website that tells you all about it. So thank you.
Questions for Elinor? Don't be shy.
I was just curious, when Alison was picking Dobermans, why did she pick Dobermans instead of picking a breed of dogs that was bred to fetch things, like dogs that involve fox hunts?
Now, that's a really interesting question, and people do ask me an awful lot how we decide which traits and which breeds we're working on. And the really honest answer to that is that we need to get enough dogs that have that trait. And so we had this collaboration going on with Tufts where they'd been studying the Dobermans. They had a very good way of deciding which Dobermans had OCD and which ones didn't. And they were able to give us, we need at least, we got really lucky with that trait that we found that really nice peak with 90 cases and 60 controls. With most of our things that we work with, we need at least 100 to 200 cases and 100 to 200 controls. So it's really, whichever breed we can get a really well-defined trait within a breed where we can get that many samples. And we're willing to, I mean, because there's so much stuff to be found.
I'm going to run around, I can tell. Okay, put your hand back up so I can find you again now that I'm on the other side. But we're recording this so it helps us, so that's good.
How can you tell if your dead white mouse is deaf?
That's a good question. They don't die.
He didn't answer?
No, those particular mice don't die. They die as like babies, so they are actually born. They can't apparently test them. But it's a really good question. I mean, I don't know how you would know.
I think they probably looked at the ear anatomy.
I was wondering if there is such a thing as a glutamic reuptake inhibitor that already exists.
That's a good question. As far as I know, no. And I think it's something that they're really interested in. I think it's, you know, I think that it's something that they're going to be working on developing, but they don't have yet. And the interesting question. Well, and that's an interesting question because as with a lot of things, glutamate signaling is actually very important in other parts of your body. It's important in your bones. It's important in, you know, other organs. And so the question would be, how do you design a drug that targets those receptors in the brain without affecting all the other functions of those genes? But I think clearly the fact that our work, the work in human OCD and work in a lot of other human psychiatric disorders is pointing towards these synapses as being really important, that it's something that we want to look at.
I thought it was a great talk. Thanks.
Thank
That's
you.
one thing. The other is that I just want to get something clarified. In humans, a lot of these things doesn't seem to be caused by inbreeding, whatever that is. But with dogs, when you have these purebreds, you seem to get a whole suite of disease tendencies,
Yeah.
cancer, heart disease, I don't know, all these kinds of things that you wouldn't get if nature could just select naturally. Do you attribute this to the inbreeding? In other words, one way to avoid these things is to stop the hyperbreeding.
I think that, yeah, no, I think that is one of the great conundrums with dog breeds because you can look, it turns out that there's over 400 dog breeds worldwide. And, you know, personally, I'm totally fascinated by them. I think it's amazing that we can create that kind of variation. They have particular behaviors in the breeds that people really love. And it, you know, I would hate to say that there's something wrong with all of the dog breeds. But at the same time, you're right. The process of creating a population with very little genetic diversity puts you at a much higher risk of having these kind of inherited diseases. And, you know, whether, you know, you kind of think of it, you could take a group of, you know, 1,000 humans and you could count up all of them that get some kind of disease and it might be quite high, but it's just not all the same kind of disease. And what you're seeing in the dog breeds is that you're getting, rather than getting a whole spectrum of disease, you're getting a particular one that's affecting a lot of dogs in the breed.
Elinor, I just wanted to follow up on that and ask you to talk about two different things that are related. One is more inbred human populations and what you're seeing there, whether there's also peaks of disease. And the other is the relationship between using kind of inbreeding and genetic disorders that you can find things in small numbers to help you understand what might be more random things, other things that happen in humans that you wouldn't see as a familial disease, but happen in more sporadic mutations?
Yeah, no, those are both interesting questions. I'm probably not qualified to answer the first one. I don't know that much about inbred human populations. There's definitely been a lot of genetic work done over the years with those populations, but I don't know as much about that, certainly not as far as psychiatric diseases go. But as far as the second question, I think that dogs are kind of inhabiting this middle ground. For years, we've been trying to do disease research in mice, and we've learned a lot. But it just has certain limitations by the fact that we basically have to have an idea beforehand of what we're interested in, go and change that in the mouse and see what the effect is. In dogs, we do have this beauty of having just naturally occurring mutations that are very high frequency in certain populations. It's possible that some of those mutations would be more severe than what we might see in humans, where you have a lot of different kinds of things going on. But I think it is going to get closer to the actual mechanisms that have gone wrong in humans. And I think that it gets back to a fundamental point of all of this genome-wide association mapping stuff, is a lot of people say, well, what are you doing? You're going to find the gene, and what will you do next? Are you going to go fix the gene? And I think that's actually not really the point of this. The point of finding the genes is not to be able to fix the DNA itself. It's to understand how do you get to having the disease. So with the cancer work we do, by the time you get the cancer, you might have a lot of changes in the DNA that actually makes the cancer, but by looking at a dog breed that gets cancer, you can actually say, what's the first change? What's the change that they inherited from their parents? What went wrong before everything else happened? And what's that kind of underlying mechanism? And I think that's what we really want to start to use dogs to try and get at, to actually come up with those more targeted therapies.
You said that some of the breeds have diseases that were maybe caused by traits that they were selected for, like dogs that needed to be obsessed with things in the task they were given would maybe develop OCD or something like that. But are there some of the breeds seem to have physical potential diseases or high rate of certain diseases where it's hard to see what the benefit of it was? And was it just sort of random, or what is sort of known about that?
That's actually a really interesting point. And I think that with the behavioral stuff, we have a better hypothesis that it's related to selection because we know that there was selection on behaviors. But even then, it's actually quite hard to know for sure that it was selection that was driving it. I mean, what causes the high rates of disease is that you have some kind of risk-causing mutation in the population that for some reason becomes very high frequency in that breed. And there's two ways for that to happen. It could be random chance. It goes through this population bottleneck when the breed gets created and that mutation happens to come through and get passed on to all the dogs. That can happen by selection. You can decide that mutation is doing something good and push it up, so it's very common in the population because you're favoring it. And genetically, those two things are actually quite hard to tell apart. And so as far as how much our selecting on behaviors is actually underlying the psychiatric disease, we can't say right now. It's still much more of a hypothesis than anything we've proven.
Is there a difference between compulsive disorders and stereotypies that we might see in animals that, say, are in captivity, that develop repetitive behaviors that may or may not be associated with the genetic SNPs that you found?
That's an interesting question. The short answer would be I'm probably not totally qualified to answer that, but I'm willing to give it a try. I would suspect that the behaviors that you're seeing in captive animals are a response to some kind of anxiety that's induced by the environment they're living in. And it may be that what we're seeing on a genetic level is that rather than having an anxiety induced by the environment, you're having an anxiety induced by your genetics. But you're then using the behaviors as a way to try and control that anxiety. And so it may not be the behaviors itself that are the critical piece, but the anxiety that precedes it.
Good speculation. Other questions? Students in the audience? Oh, wonderful.
I have a question on your guessing on digestive system of the dogs. There was a myth in early studies of genetics of dogs that if you feed the dog only meat, they would develop immunity to, I forgot the name of the disease in English, the typical dog disease that everybody gets immunized?
I don't know anything about that. But there's probably somebody in this audience who might. But yeah, no, I think it would be an interesting question. Clearly, they've adapted to our diet, but whether that had a cost associated with it, it would be interesting to know.
Interesting. Other questions? Okay, well, if there aren't any other questions, please stay. We have a reception outside, lots of food and beverages to drink, and please join me in thanking Elinor Karlsson.